Good morning, students. Once again, we are going to discuss the new topic photosynthetic pigments absorb the light that powers photosynthesis. In this topic, we are going to discuss about mostly chlorophylls, right? Chlorophyll A, B, carotenoids, and what is going on inside the thylakoid membrane. So, this topic is very important because when the pigments absorbs light, they get powered, and this power is transferred to prepare glucose molecule. So, let us start with this topic with the help of book. So, our topic is photosynthetic pigments absorb the light that powers photosynthesis. The energy of sunlight is first absorbed by the pigments of the plants. All pigments active in photosynthesis are found in the chloroplast. Structure and absorption spectra of several photosynthetic pigments we will see. The chlorophyll and bacteriochlorophyll are the typical pigment of photosynthetic organisms, but all organisms contain a mixture of more than one kind of pigment, each serving a specific function. मतलब कि सिर्फ क्लोरोफिल ही नहीं है जो फोटोसिंथेसिस में रोल प्ले करते हैं, बट अदर lots of molecules are there which together in a coordinated form play important role and finalize this process. So, chlorophyll A and B are abundant in green plants, and C and D are found in some protists and cyanobacteria. All chlorophylls have a complex ring structure that is chemically related to the porphyrin ring, porphyrin like groups found in hemoglobin and cytochrome. जैसा हेमोग्लोबिन एंड साइटोक्रोम में स्ट्रक्चर होता है रिंग लाइक, वैसा ही क्लोरोफिल में भी रिंग लाइक स्ट्रक्चर होता है. इन एडिशन लेकिन एक और अंतर क्या होता है, ए लॉन्ग कार्बन हाइड्रोकार्बन टेल इज ऑलमोस्ट ऑलवेज अटैच टू द रिंग स्ट्रक्चर. एक हाइड्रोकार्बन लंबा सा टेल रहता है. वो टेल क्या करता है, द टेल एंकर्स द क्लोरोफिल, पकड़े रहता है उसको टू द हाइड्रोफोबिक पोर्शन ऑफ इट्स इन्वायरमेंट. हाइड्रोफोबिक इन्वायरमेंट में उसको पकड़ने में, एक पकड़ देने में, मजबूती देने में वो टेल जो है वो हेल्प करते हैं. द रिंग स्ट्रक्चर कंटेन्स सम लूजली बाउंड इलेक्ट्रॉन्स एंड इज द पार्ट ऑफ द मॉलिक्यूल इन्वॉल्व्ड इन इलेक्ट्रॉन ट्रांजिशन एंड रिडॉक्स रिएक्शन. ये जो रिंग स्ट्रक्चर है क्लोरोफिल का, उसके साथ कुछ हल्का लूजली बाउंड इलेक्ट्रॉन्स होते हैं जो जैसे ही सनलाइट पड़ता है वो इलेक्ट्रॉन एक्साइटेड होते हैं और मूव करना शुरू होते हैं और हमारा फोटोसिंथेसिस का प्रोसेस शुरू हो जाता है. द डिफरेंट टाइप्स ऑफ कैरोटीनॉइड्स फाउंड इन फोटोसिंथेटिक ऑर्गेनिजम्स आर ऑल लीनियर मॉलिक्यूल्स with multiple conjugated double bonds. Absorption bands in 400 to 500 nanometer region gives carotenoid their characteristic orange color. The color of carrot is due to carotenoid, that is beta carotene, whose structure and absorption spectrum are shown. Carotenoids are found in all photosynthetic organisms except for mutant incapable of living outside the laboratory. Carotenoids are integral constituent of the thylakoid membrane and are usually associated intimately with both antenna and reaction center pigment protein. So, carotenoids दोनों जगह पे होते हैं, antenna और reaction center दोनों में. The light absorbed by the carotenoids is transferred to chlorophyll for photosynthesis. कैरोटीनॉइड जो लाइट एब्जॉर्ब करते हैं वही लाइट क्लोरोफिल को मिलता है और यहाँ से फोटोसिंथेसिस शुरू होता है. बिकॉज ऑफ दिस रोल दे आर कॉल्ड एसेसरी पिगमेंट्स. सो यहाँ पे हम देख सकते हैं कि किस तरह से कैरोटीनॉइड एंड क्लोरोफिल का एक साथ टू ऑल टुगेदर वर्क है. यहाँ लुक हियर, दिस इज द graph representation of and numbered well. So we can see here absorption and wavelength. At y side it is written absorption, light absorption, and here wavelength, which type of wavelength is absorbed by which type of molecule. The colors represent different types of molecule. So one number, which is black, look here, black is representing bacteriochlorophyll. So I have colored different according to their graph. So black color is bacteriochlorophyll, green is chlorophyll A, red is chlorophyll B, and blue is beta carotene, and purple represents phycoerythrobilin. So all four are colored and the names are also colored according to their graph. So look, the highest peak is given by chlorophyll. The highest peak is of red color, which represent chlorophyll B molecule, and between 400 to 500. So this is almost the blue region, blue color region. Look here. And in the blue color region, chlorophyll B molecule absorbs highest amount of light. And in also red region, but it is lower, right? So chlorophyll B molecule become active or highly active in blue colored wavelength of light, which is 400 to 500 nanometer range. And the next one is green colored, which is chlorophyll B. We can see here chlorophyll B molecule is also active, but it is mostly active among between the red region of the light. So approx 680 to 700 nanometer of the length, and it is also active in blue region. The next one is about purple light, which is phycoerythrobilin. Phycoerythrobilin is active between 500 to 600 nanometer, and the blue, which is beta carotene, we can see it is also active. Blue beta carotene is active between 400 to 500 region only. It is not active even single here. So it is only become active in blue region of light, and it reflects all light, so it looks like orange colored pigment. So beta carotene represents orange color after reflection, and the black one represents bacteriochlorophyll A. It works below 400 nanometer of the region and higher than 700 to 800 nanometer range of the light. So bacteriochlorophyll works, its range is much more than chlorophyll A and B and beta carotene. It is highly active below 400 nanometer of the range, and after 700 nanometer of the range it become highly active. So that is infrared region and UV region. So this here, the absorption spectrum shown are of pure pigment dissolved in non-polar solvent except for curve 4, that is phycoerythrobilin, which represents an aqueous buffer of phycoerythrin, a protein from cyanobacteria that contains a phycoerythrobilin chromophore covalently attached to the peptide chain. In many cases, the spectra of photosynthetic pigment in vivo are substantially affected by the environment of the pigments in the photosynthetic membrane. So, we understand this graph, how chlorophyll A and chlorophyll B become active in which type of wavelength of the range. So student, we understand here that action spectrum, or we can say absorption spectrum of both chlorophyll is different, and even carotenoid is also different. So, and as we have studied about the structure of the molecules, so we can once again we go through the molecular structure of photosynthetic pigment, and after that we will see give a little look to the chlorophyll molecule structure. So molecular structure of some photosynthetic pigments, that is A number, the chlorophyll have a porphyrin ring like structure with a magnesium atom coordinated in the center and a long hydrophobic hydrocarbon chain tail that anchors them in the photosynthetic membrane. The porphyrin like ring is the site of the electron rearrangement that occurs when the chlorophyll is excited, and of the unpaired electrons when it is either oxidized or reduced. Various chlorophylls differ chiefly in the substituent around the ring and the pattern of double bond. Carotenoid are linear polyene that serve as both antenna pigment and photoprotective agent. Bilene pigments are open chain tetrapyroles found in antenna structure known as phycobilisome that occurs in the cyanobacteria and red algae. So, figure 7.6 represents different types of molecule. We can give a look there. So, give a look here. First one is chlorophyll A. Look, a long hydrocarbon tail. It helps them to anchor in hydrophobic region, and this is a ring like a structure which is light green color called porphyrin ring, and in the middle of the ring is magnesium. But the difference between chlorophyll A and B, all structure is similar, but only the group, which is in chlorophyll A it is methyl group, but in chlorophyll B it is aldehyde group. Rest all even each and single atom and electrons are similar in chlorophyll A and B, but only this alkyl group, that is methyl group, is different, and it is aldehyde in chlorophyll B. And in bacteriochlorophyll, even all structure is similar, but you can give a look here. Here methyl group is present, but here something different types of structure we can see here. That's why bacteriochlorophyll become able to absorb light of below 400 nanometer up to the 800 nanometer of the range. So this is the structural difference among these three different types of pigments. And look, in section B we can see carotenoids. How carotenoids contain two types of ring structure and all double bonded structure. This is beta carotene, and here we can see bilene pigments. So the structure is here phycoerythrobilene. It is mostly found in cyanobacteria, and that's the end of this video. So hope student we have learned about different pigments and their structure and how they get powered. So in the next video we will discuss this powered chlorophyll molecule and powered and excited chlorophyll molecule, how it releases electron and where these electrons perform their activity. So thank you for watching.